All right, so here we are, and we're going to be going through reproduction for HL part 2. It's the second half of 11.4, and in this lesson we're going to be talking about fertilization and how that occurs, implantation, and how that could use for pregnancy. We're talking about hormones again. HCG is a new hormone we're talking about, and the function and form of the placenta. We're talking about other hormones, estrogen, progesterone again, and how they come back in the case of the placenta. We'll talk about more hormones for birth, estrogen and oxytocin. So you want to have your hormone table to fill out for this lesson. And application, looking at graphs, the correlation between animal size and the development of young birth at birth for other mammals. So looking at different data on that to analyze as well. Great topic too, kind of question to ask. So after we finish this lesson, guys, we'll be done topic 11, which is super exciting. So here's the targets. I can describe the steps of fertilization, and I can define key hormones involved in the reproduction and birth. So first thing first, fertilization, despite what a lot of misconceptions state, it happens actually in the fallopian tube. It doesn't happen in the uterus. A lot of people think of where it happens, but the spermatozoa have to swim all the way from the vagina through the ovary into the right fallopian tube. And it actually doesn't take a single spermatozoa, it takes several to accomplish the process of fertilization, but only one of those will actually pass on its DNA to be the father of the offspring. Once they reach the ovum, they begin to penetrate the outside cell layer, which of course the outside cells are called follicles, so the follicle cell layer of a secondary oocyte. And that secondary oocyte, remember, still is not completely haploid. We have to release another polar body still, and enter into what's called the zona pellucida. So if you look on this diagram, the pinky part, that's the zona pellucida. Then what happens is the acrosome reaction begins. So remember the acrosome from the structure of the sperm. The acrosome is the little packet at the head of the sperm that's full of enzymes. And those are enzymes that are going to react with the zona pellucida to then enable fertilization to happen. So the enzymes are going to be released, and it passes through the membrane of the ovum, and then you can see, we'll look at a bigger version picture in a second here, and you can see how the first chromosome that gets through will actually fertilize the ovum. So it's not about being the first one to the egg, it's the first one getting through the layers of the zona pellucida towards the egg. Okay, and then you can see here what's happening is we're getting kind of a layer here outside the plasma membrane of the egg to actually prevent multiple fertilization. So we'll look at that in a second as well. So here's a really nice diagram, and if you're taking digital notes, I recommend you find this one online because it's a really good diagram to show this. And what we can see here is we can see that we have the sperm making contact with the egg as step 1. Okay. Right here. So the sperm makes contact with the egg, and then we begin having the acrosome reaction. So the acrosome here at the tip of the head of the sperm is going to open, and the enzymes inside are going to be released, and those allow the zona pellucida to be broken down. And then it goes into getting the cell membrane, the plasma membrane of the ovum. The photoreceptors here are going to react, and the membrane of the sperm and the egg are going to fuse, since they're both phospholipid bilayers, and allow for the sperm nucleus to enter. Now what's happening that's really interesting here is as this acrosome reaction is happening, these little purple dots, okay, these are protein receptors, and they're going to then notice the fact that the sperm is there, and that's going to cause these cortical granules on the outside, but inside of the plasma membrane, to fuse with the nucleus and create this barrier. Okay, it's called the vitelline layer, and this barrier here in this space from the zona pellucida into the plasma membrane is going to block any further sperm from being able to fertilize the egg. So one nucleus inside the egg, one nucleus from the sperm together is going to make a fertilized egg. Okay, so the two membranes are going to fuse, and only the DNA of the sperm enters the ovum, the cytoplasm. Nothing else enters. The tail, the mitochondria, everything else stays on the outside. And when we fuse those two sets of haploid chromosomes, that's going to lead to a diploid zygote. Remember, the zygote is a single-celled organism of fertilized egg. It hasn't developed or divided yet. It's just a single cell, but it has half DNA from mom, half from dad. If we get more than one chromosome trying to enter the ovum, it's what's called polyspermy, so poly, of course, meaning multiple. This leads to too much DNA, and the zygote will actually fail. This is not how we want it to happen. So how we prevent that and from wasting a fertilization event, and as far as reproduction is concerned and evolution is concerned, we want to have successful reproduction to make the next generation. Once spermatozoon's membrane fuses with the ovum, it's going to then cause its cortical granules we talked earlier to release chemicals, which makes zona pellucida impermeable. So it makes it like a shield around the entire zona pellucida, so no other sperm can then enter. So again, look at the steps here. And if you don't have this diagram, you want to put it in your notes for sure. Okay, I'm going to give you the steps for this process in a jumble, and then I want you to put them in the right order. So here are the steps in the incorrect order. I want you to pause the video, take a minute, and put these 7 steps in the correct order. So pause the video and go for it. Okay, so here we go. First step is sperm are attracted to the secondary oocyte by a chemical signal. So how the sperm know which fallopian tube to go to is based on this chemical signal. The first sperm reaches the pellucida, binds, and releases the contents of its acrosome. And so the acrosome digests a path through the zona pellucida for the sperm to the plasma membrane. And then that fuses with the secondary oocyte's plasma membrane, which causes the cortical reaction. The membranes fuse that triggers the shield kind of forming. Cortical granules inside the oocyte fuse with the plasma membrane and release their contents. And that enzymes from that cause the pellucida to become hard, preventing polyspermy. And the 2 nuclei are going to fuse inside the egg, and that's going to become the zygote, and the nucleus of the zygote in particular. So what you should take now is go back to your diagrams, your antidiagrams you did last class, or first half of this lesson, and I want you to finish annotating them, especially focusing on the spermatozoa and on the ovum, the egg, and really talk about the function. Why do we have the follicle cells around the egg? Why do we have the cortical cells? What's the purpose of them? Talk about how we prevent polyspermy as part of your answer. And you can also take a minute and add some more details to your female reproductive system diagrams to talk about. It does show fertilization on there, and you can talk about that and show that as part of your notes. You definitely should. So pause the video and go ahead and do that. Take about 10 minutes to do so. Okay, key idea at this point in the lesson, so go ahead and give it a read. If you have any questions, ask a classmate, ask your substitute teacher, ask me. Now we're going to talk about implantation. So implantation happens after successful fertilization, and what's going to happen is after the zygote is formed through fertilization, it's going to immediately begin doing mitotic division, so dividing from a diploid to a diploid, making identical cells. And it's going to do this as it's being moved through the fallopian tubes. You remember from talking about the fallopian tubes, we have fertilization happening here. The fallopian tubes have these kind of finger-like projections, or cilia, that move the egg or the ovule through them towards the uterus. They're quite gentle because the ovum are quite delicate. And once it starts to divide, it's now an embryo. Okay? So as soon as it becomes multicellular, it's called an embryo, and it's going to make its way all the way through the fallopian tube into the uterine wall for implantation. It takes about 7 days to get there. And once it gets to the 7th day and it's at the uterus implantation, it's now about 100 cells in size, and the name for that particular level of embryo development is called a blastocyst. It's an important point in development. Now, for this to happen, for implantation to work, the uterus has to have this lining here. So you can see the uterus, the walls is kind of like paler pink color, and there's like grayish pink around here. This is the lining that is built up. And every month, the woman's body builds that lining in preparation for implantation. And if she does not get pregnant, then that lining is shed through her period, and we try again the next month. So the blastocyst has to implant into the lining, and it has to do what's called highly vascular, lots and lots of blood vessels, so we're able to support the embryo and then the fetus and to build the placenta through that. So look at the blastocyst here. So blastocyst, which again is a 100-cell level of embryo development, it's already begun some very early differentiation. And we can look at this picture here. We can see the trophoblast at the end here. it's going to end up forming the fetal side. So we look here, the pink part, that's the fetal side of the placenta. And the inner mass, the blue, is going to become the embryo itself. So we have the placenta part, we have the embryo itself, all this differentiation happening really early. It also has the fluid cavity, which is right here, the white part, and that's kind of where the embryo will develop. That's the cavity inside of the uterus. During these very first few days after fertilization, before the placenta has fully formed, the embryo is going to start dividing. But it's not going to get very big. It's going to start dividing the cells smaller and smaller and specializing those cells because there's not a lot of nutrients available to me. The nutrients that were already in the fertilized egg are all it has until the placenta arrives and can give it new nutrients. So it's limited in its size because of that reason. So you want to go back to this diagram here and add information to this part. This is what we talk about now for fertilization and early differentiation and development. Okay, let's talk about the placenta. So the placenta is a structure that's what connects the mother to the embryo and our fetus. It has two parts. It has an embryonic side and it has a fetal side. So this umbilical cord connects to the fetus, the fetal side of the placenta, and here we have the maternal side of the placenta. What is the purpose of it? What does it do? Well, everything that the fetus and embryo need, nutrients, etc., thing it produces, get rid of, like waste, all has to be dealt with through the placenta. So it's an exchange place for nutrients, waste, oxygen, carbon dioxide, hormones, water, urea, vitamins, minerals, all those different things. And these two, the blood of both parents, mother and the fetus or embryo, don't mix. What happens here is we have the different structures that help us to happen. So take a look at this. What do you see in here? Think about topic 6 and the circulatory system. Well, here we have these capillaries. Okay, these are fetal capillaries coming off the umbilical vein and arteries, and they are going to be maximized for absorption and exchange of things like gases and nutrients and waste, and they're bathed in the mother's maternal blood. Okay, we have the mother's blood vessels here, her venules, arterioles, small blood vessels as well to bring things to the fetus. Now, is the blood actually mixing? No, because red blood cells don't go between. Everything that goes between enters the capillary is going to be things like waste, nutrients, hormones, water, those kinds of things, not actual blood itself. Now, if the mother smokes or drinks or takes medicine, what's going to happen with that? Well, if it's in the mother's bloodstream, it can end up inside the fetus or the embryo. So this does not prevent nicotine from entering or alcohol from entering, which is why in a lot of parts of the world, and it's very frowned upon to allow parents, mothers to drink, smoke, take medicine, that kind of stuff when they are pregnant, because it affects the placenta and therefore it affects the embryo or the fetus very strongly in a way they're not able to prevent, and they can't filter it out anyway. So again, here's your key ideas. Take a minute, write them down, check if you have any questions. Okay, let's start talking about hormones. So you want to go back to your table that you had and use that. So hormones we're working on here is 3 main hormones for post-fertilization, and those are HCG, which is secreted not by a particular gland, but by the embryo itself. And that's going to help maintain the corpus luteum, which then will secrete progesterone. And you can see here HCG peaks really early and after pregnancy and then really goes down. And this is the hormone that pregnancy tests detect, which we talked about in topic 11 with monoclonal antibodies. We have progesterone, the blue one here, being secreted by the corpus luteum, and the placenta itself also secretes them afterwards, and it's going to help maintain endometrial lining and vascularization, all those blood vessels in the uterine wall. And then we have estrogen, our purple one here, slowly increasing as time goes on, and this is going to help stimulate uterine muscle growth because, of course, the uterus has to contract to push that baby out. And mammary glands produce milk production. So it peaks at the end of pregnancy because those things are more required at the end of pregnancy, not the beginning. So take a minute and explain to a partner why the graph levels change in that way. Okay, so you should be noticing here we have HCGs really peaking at the beginning of pregnancy when you just have the early embryo because it secretes its own HCG. And then we have low progesterone at the beginning, and they're increasing over time. Progesterone decreases at the end of pregnancy because it's not needed as much for the placenta. At the end of pregnancy, you don't want the placenta anymore. You're going to get rid of it as well, and the lining as well. At 38 weeks, the fetus is pretty much fully formed, so it's going to just need to be pushed out, which is why we still have lots of estrogen with muscles and mammary glands. Okay, so parturition is the process of labor, the official name for labor, and it begins with these low-intensity contractions that are infrequent. And a lot of times women say that, and I have not been pregnant or had babies, so I'm not sure what this feels like myself, but it has gradual and kind of infrequent contractions that feel like period cramps, and then they slowly become more frequent and more intense over time. And that's your uterus contracting to be able to push baby out. The goal is to dilate the cervix for baby to pass through the birth canal. So if we look at this image here, this is the cervix, and it's pretty closed, but it needs to open up to let the baby all the way out. And that's the contraction going to do, is to help to dilate the cervix and open up everything up so we can pass the birth canal. Throughout pregnancy, when estrogen is doing this thing, it's not only going to help make the uterine wall stronger, it's also going to create a larger number of oxytocin receptors in the uterine muscle. Remember, oxytocin is a hormone. Using a lot of those receptors, so that way oxytocin can then react to the muscle properly. At birth, what's going to happen is the pituitary gland, posterior pituitary gland in particular, is going to release oxytocin, the hormone, which then begins this feedback loop, which will continue until birth happens, and the uterus will stop contracting. There's nothing for it to contract against any longer. So we have oxytocin after the head of the baby is pushing against the cervix, and then that sends impulse to the brain. From the cervix to the brain. The brain tells the gland to secrete oxytocin. Oxytocin goes to the bloodstream to the uterus, and that tells the uterine wall to contract and push baby towards cervix. It pushes the cervix, it sends the nerve impulse, it secretes oxytocin, the same thing again and again and again. So what kind of feedback loop is that, guys? That's right, positive feedback loop. And in general, positives tend to be for extreme events. Okay, here's note-taking strategy to check and fill in those new information about different hormones. So we already had estrogen and progesterone before. Those are SL hormones as well, but you have more information now on how they relate to pregnancy and labor. And these two are the HL only ones that are new. The rest of these you should already have filled out from your SL lessons. Okay, so take a look at this graph, and we have the length of gestation or pregnancy and the body mass of all these different organisms. So what do we notice? What trends do we see? Pause the video, talk to a classmate. So what you should see here is you can see that as gestation length increases, so too does body mass at birth in general. Humans are over here. We're a little bit of an outlier. We have a longer just period compared to our body mass, and a big part of that is we're developing our brain, not just the physical body. Okay, key ideas, here you go. Go ahead and write those down, check them. And that is the end of 11.4. You are done. So well done. Take a minute and have a celebration. Topic 11, all done. Well done, guys. Hope it's helpful for you, and have a good one.